you and
Yes.
earlier this morning, I sent you an email with my videos in it.
Oh, okay.
Do you have, or should I send it to Tom?
Um,
Who's got a good,
send it to Stan Bissinger.
say again?
Stan Bissinger. Bissinger.
He's not here.
Well, he's coming.
Okay, but I don't have an address for him.
Well, let me give that to you.
Hmm?
Um, I can give that to you
Okay,
in a minute. Um,
I've got
okay.
a random bit.
okay.
Um, the Russians fired an ICBM at Ukraine, an experimental ICBM to Ukraine, the city of Dnipro, which is their rocket manufacturing and testing location in Ukraine. Dnipro. I can't say it right.
Yep.
Anyway, we've got video footage of this. It came down at a high angle of trajectory, and so this is the first time an ICBM has ever been fired in warfare.
Well, it's hardly intercontinental
When it
from
went
Yeah,
from
but
Russia to
Moscow
Ukraine.
to
it
Ukraine.
still
This is
is an
true.
escalation, and it happened a couple days ago. Yeah.
Right.
It's an ICBM because it went into outer space before
Yep.
it came down
Yep.
to the. It's not. It was an intermediate range, meaning they've got a maximum range of about 600 or so miles, as opposed to thousands of miles. But this is something we should be concerned about. We shouldn't make light of it, if you ask me.
Jason, did they also tell you that they warned the United States and NATO about an hour before it went up?
As we usually do for them whenever we set up a satellite.
For good reason, because you don't
Very
want
good.
your potential opponent to nuke you just because you send up an ICBM.
Well, it's in the interest of both parties that we don't
But we are not a party to that particular war, supposedly.
Anytime you see the rockets light and the missile go up, they don't know for some time the exact trajectory. You don't
Yep.
want to,
Yep.
Yeah.
you don't want NORAD to get the wrong ideas.
You don't. Especially
They're
if you're
saying
Russia.
they're also saying that the U.S. has no defense against that particular type of missile.
Probably
you wouldn't
not.
need because it's 600 miles. You can't hit anything outside of continental Europe with just 600 miles.
That particular one.
But continental Europe would be more than enough to flatten Russia right now. Which is why they warned a couple of people an hour before it went up. Or before it went up.
Here's a question. If Russia was to launch a single nuke against an adversary, would a single nuke be sufficient to trigger an all-out hundreds or thousands of missile response? Or should we do a more proportional response of, say, if they fire one missile, we fire, I don't know, one or two of our own?
Five.
Yeah, proportional is likely, I think.
We should
That still could easily escalate.
Right. Not thousands of missiles in response to one.
Right. That's why I said proportional is likely, but it's dangerous. It could easily escalate from there.
Yep.
If you would think back to the Manhattan Project, they were considering doing a demonstration firing or bombing, but there wasn't any real convincing way to know that there would be a sufficient impact to make a difference. So that question has been asked before.
Now,
I don't know.
you had said we
I
had
don't know why. I was just say I don't know why anyone hasn't brought this up before on TV or otherwise, but with all this conflict going on in Russia versus Ukraine, I wonder if it's time to consider global abandonment of the nuclear non-proliferation treaty.
No,
Because-
it's time to
Oh,
consider
no,
abandonment
thank
of
you.
nuclear
Yeah.
weapons.
No, thank you.
H-Time,
Okay.
you had said we had...
You were
We've
asking.
We've already abandoned many of these treaties. They've either expired or we've pulled out of them.
Yep.
So that's a moot point. It's already many of the SALT and START treaties that we've negotiated in the past have either expired or we've pulled out of. We've undone much of the progress that we've made in prior generations under the Reagan administration and the Carter administration and whatnot. If you look at we've got a fraction of the war we had.
Yes.
Is when we had over 10,000 warheads just on each side.
We won't need anything in the future. When Trump is in, there won't be any war.
Yeah,
war has
and not only,
Okay.
and basically Russia's way of
all 24 hours.
reducing their arsenal has been to export it to other friendly countries to Russia. So,
Mr. Walsh, you asked.
I find it
Yeah.
reassuring that we've got a president that's peace and is striving his best to make peace and is clearly in. I mean, in these times of international trouble, it's the current administration that's escalated the war. So you all laugh, but it's the Biden administration that has ratcheted up the war to a new level. So that's kind of hypocritical to kind of laugh at that because I personally want.
He's
it's
restrained
after
him.
10 o'clock.
He's always been 6 months behind.
H-Time, I want to...
I'm waiting for Carole to post some videos up on the chat. Is that something that we're going to do, Carole?
I have done that. Can you look in chat?
Well, maybe I came in late. I don't know. It might probably.
No, I just did it like a minute or two ago.
I can't see it either, Carole.
Chat is empty.
I'm not seeing
Did you
anything,
actually
chat.
hit the send, Carole?
No, I just copied it over.
H-Time, you said we have no defense against the kind of missile that they sent. I know we don't have defense against the hypersonic missiles, but this wasn't one of those.
We've got the interceptors in California and Alaska.
There's a problem. How can I say this? If you can see a bullet coming at you, whether it's from a 22 or it's a nuclear tipped, whatever, if you can see it, then you sometimes can do something about it. In the case of a nuclear weapon, you don't know. Perhaps if you interfere with it, it will detonate in place. So instead of landing in Portland, it will detonate 200 miles over the Aleutian Islands and do horrible things to the fishing industry. You don't know.
Can you see that now?
Yeah, I got it.
Okay, good.
These are not things to play with. And if they actually sent I recommended a book last week.
Yeah, well, Ukraine had that philosophy in 1994 when they decided to give up. They were the third largest nuclear armed country, and they decided to give their nuclear weapons up. So maybe since that hasn't worked out too well for them, maybe They ought to covertly arm them in the lame duck with nuclear weapons.
A
And then
better,
the
a
administration
better
comes in and say,
solution.
Every nuclear weapon has a fingerprint on it. You could trace it back to the reactor from which the core material was taken from. So you could trace back
True.
from the explosion which country produced the nuclear bomb after the bomb has gone off.
So maybe not covertly, just announce that we're selling it to them, and that we've now washed our hands of it since the new administration wants to get rid.
Okay, it's 5 after 10. We should move on to the presentation.
There's a
And we
reason
should
why.
also raise our hands rather than talking over each other.
Well, it's a little different for during the pre-presentation period, but it's time to move on to the presentation now.
Okay, Stan, you can see that in chat now.
Yeah, I've already copied it into Word, so I've got it.
Oh, okay. Then,
For anyone who doesn't know, Carole Ashmore is our presenter this morning.
let's see.
And the topic is protein folding, right?
Yes,
And the pluses and minuses of it.
yes. Or, as it says, predictions and perils. If I can just share this.
Now, Carole, as you do the presentation, are there times when you will take Q&A, or how do you want to do that?
Okay, let me see if I can get this shared out first.
Okay. Now, the
Can
question
you
is,
all see computer-aided
yes,
protein folding?
yes.
Yep.
Predictions and perils.
So at what point are you going to want to take QA? Do you have certain spots during the presentation, or you want to do the whole thing
Well,
first, or what?
I have 5 videos that I am going to show. And I would say I would take questions after each video.
Okay. If you have a question, I think everyone knows you use the reactions button to raise your hand. And we'll try to take you in order.
Okay, it's being recorded and you can all see this, right?
Yes.
Okay. Now, going to do just a really quick summary of organic chemistry because that's what we're into. Don't panic. Okay, basically, organic chemistry is carbon chemistry. And it mostly has to do with covalent bonds rather than electrovalent bonds because carbon atoms bond covalently, meaning that rather than 2 elements losing an electron and gaining an electron and crashing together with a heavy electronic bond because they are different, one's positive and one's negative. Covalent bonding is a little friendlier, a little squishier, and it just involves the carbon atom sharing some electrons with the outer shell of the next carbon atom or the hydrogen atom or whatever it's playing around with. Covalent bonds are less aggressive and easier to rip apart than electrovalent. Okay. Carbon, you will remember, is number 6 in the periodic table. I'm kind of pointing at it here. And it has a possibility of 4 bonds. And organic chemistry basically involves chains of carbon atoms with different lengths and different geometries and various elements and radicals attached at various points. Carbon-carbon bonds are covalent, sharing electrons in a single or double bonds. Various attachments may be covalent or ionic, depending. The longer the carbon chains, the more possibilities there are for multiple geometric configurations of the same chemical formula. For instance, when you're looking at carbon chains, a 1 carbon chain is methane, and it's a carbon atom with 4 hydrogen atoms attached. And that's the only possibility. The next one is ethane, 2 carbon atoms. Again, no different possibilities. But when you get to propane, 3 carbon atoms, and you start looking at attaching things, you can attach, say if you want propyl alcohol, you can attach the OH radical to the end carbon, either end carbon, or you can attach it to the middle carbon. And you get subtly different liquids. You get propyl alcohol and isopropyl alcohol. And what you get into is the longer the carbon chain, the more different possibilities you have for folding it up, mashing it together, what have you. And, you can have exactly the same lengthy carbon chain that will fold or attach things in all kinds of different ways, because the geometry has so many possibilities as soon as you start attaching things to a 3 carbon or longer chain. Okay. For instance, now here's a benzene ring is 6 carbons, and people kept looking at benzene and saying, that doesn't have enough hydrogen atoms to fill up all the bonds. What's going on here?
Say, Carole, can you maximize it on the screen, I guess, so that we can see a bigger picture?
Okay, let's see. Bigger picture. How's that? I think that's as big as I get. And finally, I think the chemist said it came to him in a dream that the 6 carbon chain folded up, and the 2 carbons on the end were all of a sudden snuggled up to one another, and you didn't need those extra couple of hydrogens to make all the bonds come out. So, the longer the chain, the more geometric possibilities you have. And when you get to proteins, my, long chains, long chains. And the various portions of the chain have slightly different electrochemical fields. And so, you know, they'll wander past one another and fold all of a sudden because you get an electronic attraction. And so, you know, this is just a tiny example of folding. This is serious protein folding going on, with every single one of these little dots being an amino acid. Okay. Now, as I say, that's our really quickie organic chemistry review. And I assume that you all read the intro about the 3 people who got the Nobel Prize in Chemistry for 2024, basically all being comp sci people. And things are getting real interesting with computer-aided everything in chemistry. So I would like you to play this one, the number 1, which is 10 minutes long.
Carole, if I could ask you to hold up just a second.
Sure.
There's another problem with this particular Zoom. For some reason, it doesn't recognize that I'm a pro account. And so it's going to shut off in 8 minutes, 9 minutes. Just log back into the same account. Or
Okay.
maybe I can put up the other account that would last for 3 hours.
Well, this video, I'm going to have to unshare anyway. This video is going to be 10 minutes long.
Right.
So, what?
Maybe the thing to do is to go back to the other meeting, and then we won't have this problem.
I don't know what the link is for this other meeting.
Where is the link for the other meeting?
Yeah.
David, can you assign me as host?
Sure. I think I can assign you as host.
Well, you both have a pro account. I'm not sure why.
I know.
Doesn't
Just a
recognize-
second.
Why can't you sign me as host with the
You should be host, right, Tom? It's Tom.
I'm looking. Host, says host me. Yep.
Now, can you upgrade
Okay, now
this?
should we all log out and try
No,
to get
just
in?
a minute.
No.
No.
Just
Can
wait.
you upgrade this meeting, Tom?
It's since I'm the only one listed as host, remaining meeting time 7 minutes. Okay, why is that?
Because this particular meeting.
Yeah, okay. How do I upgrade?
I don't know. I don't know if it's possible.
Oh, I'm sure it's possible. I just don't know how.
Can you go to Zoom?
Well, could we have whoever's going to play the video actually play the video, and then you can be fussing around with trying to upgrade the meeting?
I'm afraid what's going to happen is they're going to shut us off in about 4 minutes.
in the middle of the video.
Yep.
Okay. Then what do we do? Perhaps we all log off and log back into a different,
Let's work the problem here, people. We've got 4 minutes to decide.
find.
Wonderful video. Thank you very much. I need to watch it again. A lot of that went over my head.
It would be nice if you posted all those videos again on this chat, because I lost it when we switched.
Okay, I can do that. Now let's see, we've done 3 videos so far.
Just list the whole.
If you go to the
DeepMind,
if you go to the DeepMind website that I posted, yeah, they have like 177 videos you can choose from. They're like that. So
yes, I
play with it.
That's a video folding problem.
Carole, I just posted
I
it.
even had, I mean, I have more videos listed than I'm using in the presentation. These basically,
I just posted them with Carole
oh, where?
in the chat.
Okay.
And I just posted the ones that we're going to watch. I didn't post all the other ones.
Right. And there are 5 total videos. The first 4, of which I think we've seen 3, are all on the folding problem. And the 5th one is on prion diseases. I keep trying to say prion because that's an alternate pronunciation that I used to use when it was strictly in my written vocabulary. And then when I heard people talking about it, I figured out that prion was the preferred pronunciation. So, do we have any questions on that video, or shall we watch the 4th one?
There's one. Chris has one.
Okay. Some new information is that it's a little bit complicated. Since you have 24 amino acids, it is a sequence of 3 nucleotides that code for an amino acid in a protein. And since there are 64 possible 3 base sequences from 4 bases, you have 20 amino acids and 64 codons. So you have more than one codon that codes for an amino acid, okay? Now, what that means is that you can have different mRNA sequences that code for the same amino acid. Everybody got that? Because there are different codons for the same amino acid, you can have different mRNA sequences that code for the same amino acid. But if you take those 2 different mRNA sequences and translate it into a protein in the ribosome in the cell, you can get different protein foldings. The reason is because the rate of translation is different. And if the protein is synthesized from the mRNA fast, it has different folding options than if it's translated slow. If it is translated slowly, the part that has already been translated to protein has opportunities. It is more under kinetic control than thermodynamic control. This is well-known stuff. It's fairly recent, but it's well-known. The same amino acid sequence, depending upon its rate of translation in the ribosome in the cell, will have different foldings. This is well known, but it's very recent. So that's an additional. This alpha fold is terrific stuff. It's great. It's going to help a lot of biology, but that's another dimension of difficulty that isn't going to have to address if it's ever going to be 100% accurate.
This is true. And timing is important for many chemical processes. The first one I ever heard of was if you produce heavy water, and that is basically using deuterium instead of the hydrogen atom. In other words, you produce G2O. Chemically, it is the same as water. But if you feed living critters heavy water, eventually, what happens is that the chemical reactions are slower because the deuterium atom essentially weighs twice as much as the hydrogen atom. And so if you make enough heavy water that you can feed it to lab mice and guinea pigs and things like that, when the percentage of heavy water in their body gets to be above, I think it's 10%, they die, because the timing on their water-based chemical processes is off. And I mean, that's a very small, it's nothing akin to the complexity of a folded protein. But yeah, timing can be incredibly important.
In protein folding, there are two competing forces. One is thermodynamic control, and the other is kinetic control. And
now, what do you mean by kinetic control?
Kinetic control means
So fast.
the rate. One is the lowest energy situation, and one is controlled by the rate, because the
How
rate
fast you
the rate-
fold the protein.
the rate of folding can
Okay.
control local co-translational folding. It gives the opportunity. If it's slow enough, it gives the opportunity for folding locally that would not occur if it went fast. That is a contribution for kinetic control, the rate, rather than thermodynamic control. And those two forces sometimes compete. So AlphaFold is great. If it wants to be 100% accurate, it's going to have to take this into control. And it's going to have to know which mRNA sequence which sequence of nucleotides and which sequence of codons is going to give that code for the same amino acid sequence. It's going to have to know which of those is going to give which protein structure. So it has, AlphaFold still has a way to go.
Oh, yes. Well,
Yeah.
at this point, they are on AlphaFold 3, which is supposed to be much better than AlphaFold 2, which was in turn much better than the original AlphaFold. So the program keeps evolving.
Right. I just wanted to, I guess my point is similar to Chris's. Yes, namely the sequence of amino acids does pretty much dictate how it folds into its final shape, but temperature, pH, the level of acidity or whatever, makes a difference and can influence the folding process
And
result in variations in the shape. So
we are very quickly coming up again on prion diseases,
yeah,
which
okay.
are misfolded proteins.
Even proteins created within a cell can end up with different shapes using the same amino acid sequence. So depending on pressure and all sorts of things affecting the cell, you can end up with a different protein, well, different shapes of proteins, which affect their efficacy and how they actually act in the body.
Now, let's see.
Eric
Have we done all 4 of the?
There's one video left.
Yeah, and Eric has his, and Eric have their hands up.
I just wanted to throw another monkey wrench into things. Try to imagine how RFK, our new future health minister, is going to preside over, understanding and prioritizing all the possible funding possibilities. And this, it's just scary and depressing. It's sort of like it would be sort of like the demagogue party had won and appointed Greta Thunberg to be energy secretary.
I have a quick question for William.
I disagree.
Kinetic versus thermal. If you have Brownian motion and that's motion, doesn't motion and thermal sort of go hand in hand? More motion makes it hotter.
Or vice versa, yeah.
Yes,
The
but
hotter produces more Brownian motion.
I thought that they went hand in hand. I mean, if things speed up, it's like if you increase the heat of a balloon, the balloon gets larger. So the scale of the balloon and the scale of the heat is proportional. It's kind of the same phenomenon, I guess.
No.
Well, it's like leaves don't make wind blow. Moving leaves do not make wind blow. So maybe it's the thermal causes the motion.
Okay. Now let's have we done the first 4 videos, which were all basically on?
I've got the next video queued up and I'm ready to go.
Okay, but are we on number 4 or number 5?
Well, we're going to be watching the 8-minute one.
That doesn't help.
says asterisk 4, parentheses, 8 minutes.
Right.
It's this gal here.
Okay. Okay, yes. This is still then on alpha folding. And then the last video is on the prion disease problem.
It's almost 11:45. Do we have time for both of them?
Well,
Possibly not.
Maybe we don't need another alpha folding thing.
okay, then
Or
Maybe
maybe
we,
you want to hold off on prion diseases for another meeting.
well, what I've got on them isn't that long. I think you're right. We should skip this video.
Okay.
I thought I had it timed, but we kind of wasted a lot of
Yeah, we
time
lost some at the
getting
beginning.
Yeah,
there.
we lost
Sorry about that.
Well, you know, just a second. I gotta get I've got a little more stuff here for introducing the,
Okay.
okay, here we go.
Eric introduced an interesting side thought.
I missed that, David.
If it's quick, go ahead.
Well, it's not quick, but
Okay, then let's wait till after this.
Okay, now at this point, we're looking at the fact that creating new proteins and new ways to fold old proteins leads to the danger of creating new prion diseases. And prion diseases are really scary because they are not created by anything that's alive. You know, you can kill bacteria that create diseases. You can kill viruses that create diseases. You can kill, spores. You can't kill a misfolded protein. And they have had cases of prion diseases moved from one person to another because of trying to transplant organs or even transplanting corneas.
Or cannibalism.
Well, yes, we'll get to that. But, you know, you autoclave the instruments you use, but it doesn't get rid. It kills all the bacteria and the viruses and everything, but it doesn't get rid of the misfolded protein that may be clinging there. Now, okay,
Ready for the video?
not quite.
Okay.
We only found out about prion diseases, back in 1920s, 1930s. And the first that we found out about was a disease called Puru. And the only people who had this disease were members of the Foray tribe in New Guinea. And these were the people that Margaret Mead and her associates, went into New Guinea and were dealing with people who had never met anyone from outside the island. And this one particular tribe had a very awkward disease that was always fatal, and it basically rotted their brains. And it turned out that they got this disease because part of their funeral processes were eating the deceased. And so they transferred the misfolded proteins that had rotted the brains of, Aunt Jenny. And when you consumed said brains during your funeral processes, you got it. Very bad. But, I mean, let's face it, it was a really easy disease to avoid. all you had to do is stay out of New Guinea and don't practice cannibalism. Later on, we got into much more dangerous diseases, prion diseases, and that was the mad cow disease epidemic. And that showed up in Britain in the 1970s. Again, 100% fatal, although sometimes there were months or years of the incubation period before the misfolded proteins manage to cross the blood-brain barrier and start rotting out your brains. And this disease was caused by a stupid mass farming practice of they wanted to wean the calves early so that their mothers could go back to producing milk for the commercial market rather than feeding the calves. And if you wean the calf too early, they don't get enough calcium and their bones don't develop right and all sorts of awkward things. So they ended up adding to the meal they were feeding to the early weaned calves bone meal, ground up bone meal to get them lots of calcium in their diet. And that was how, what is it, bovine spongiform encephalitis, mad cow disease, entered the cow population. They managed pretty much to confine it to Britain. There were like a couple of cases in Canada and one or two in the US, but they were from cows that had actually been imported from England. And once this started, people got careful about importing cows from England. And when they figured out what was causing it, of course, they quit feeding, ground up portions of your same species to the calves. And the epidemic, which at one time was up to like 1,000 infections per day in the British population, is at this point down to practically nothing. Okay,
That's good.
How about that? Are you seeing it?
Yeah.
Okay, so these are the slides I've been talking about. Sorry. And then how do we make this bigger?
Don't worry about it.
Okay.
Carole, you could try F5. I think that might be it. The F5 key. That might work. I don't know.
What, Control 5?
No, just I think it's regular F5 key.
At the top of your keyboard.
Top of the keyboard, F5, I think maximizes it. And then escape to get out. Yeah, there we go.
There, okay. So this is just a quick rundown of protein folding disease. There are things like cystic fibrosis and the neurodegenerative diseases, which aren't as bad because they're not fatal in the short term. But the actual prion misfolded protein diseases will just knock you out. And it comes from this is the unfolded protein, and it just didn't quite make it properly. And that's what causes prion diseases. Okay, let's try to share the video because it's quite comprehensive.
How many prions are there? How many different prions are there?
Lots. It tends to be species specific, but prions from other species can mess up your brain, as with the cows. But most of the times that this shows up as infectious, it's because we're doing something dumb in the farming process. You know, if they hadn't wanted to wean the calves early so that their mothers could go back to producing milk for kitties to drink, we would not have introduced the prion disease to cattle. I mean, cows are herbivores, except, when they're very young and drinking their mother's milk. So, and, it's pretty easy to get most populations not to practice cannibalism. And,
Now, when you say dumb, I'd rather say either malicious or ignorant or malevolent, because the guys in charge of these big corporations, they're not really stupid. Everything has to be thought of in terms of primary goals, and their primary goal is increased profit every quarter, not whether it's going to kill a few people like prion disease.
Well, yes.
But that negates your premise that they are not stupid.
Yeah, they're not. You give them credit there. It's either ignorant or malevolent or both, but not really stupid, I'd say.
We have to define stupid, and that's for another day.
Yeah.
What is the Nietzsche quote? Against stupidity, the gods themselves contend in vain.
Yeah.
I want it this way.
Yeah. Well, I mean, they're probably not as high IQ as the guys at DeepMind. I'll put it that way. They're dumber than them, but they're smarter than most. Way over 100 IQ. Say that. The guys that run the food, corporation.
There are two completely different definitions of dumb. Dumb can be not intelligent, and dumb can be not aware of the consequences of what you're doing.
That's what I put on when I said ignorance. They're ignorant. Yeah.
Could I ask a question, Carole?
Sure.
So prions are proteins that are common in your body.
Yes.
And are particularly common in nerve and immune cells.
Brain and spinal cord nerve tissue.
As well as the lymph nodes, which is the T and
Well, the lymph nodes are what carries it up to the brain and gets it to cross the blood-brain barrier.
Yeah, the lymph system is basically a parallel system to the blood system, is that right? And carries your immune cells. But the immune cells and the nerve cells contain the prions. But it's
Yes.
only prion disease when those proteins misfold. Is that correct?
Yes,
And
that is correct.
the
And
misfolded proteins cause properly folded proteins to misfold in some fashion.
yes,
By adjacency.
My understanding is there are kind of two levels of this. The really bad kind is where when the misfolded protein encounters a normal protein, it causes it to misfold.
Okay.
So, I mean, you're just done for.
Yeah.
With things like Alzheimer's, my understanding is that the misfolded proteins just sort of lay around and clump. They get in the way of brain functioning, but they're not just cascading and leaving holes in your brain.
Okay, so the the prion diseases and what's the name of the one you just talked about?
Mad cow disease?
No, the prion diseases are different than the degenerative diseases in that the prion diseases cascade and the degenerative diseases do not. Is that right?
That is my understanding, yes.
Okay. So the possibility exists if the, okay, and then the other question I had to do with the folding being related to the DNA. Does the DNA pattern define the folding, except that it gets screwed up when it comes in contact with a diseased prion?
Yes.
Okay. So potentially, if you could introduce properly folding prion DNA, the genetics could be that 15% that's genetic could be potentially, quote, cured by correcting the genetic DNA of the prion. Is that true? Are they talking about curing sickle cell anemia genetically?
Yeah. I mean, I watched a whole bunch of videos that I didn't, save for this.
Yeah.
And
okay.
my understanding is that with the severe type of prion disease, where the misfolded protein will encounter a proper protein and cause it to misfold. It's like dropping the ping pong ball on the mouse traps.
Yeah. It sets off a reaction. It sets off a cascade.
Yeah. It is usually from the time symptoms show up. You've got maybe a year or two before you are dead.
Yeah.
But sometimes it's years before symptoms show up because it takes it that long to get over the blood-brain barrier where it's got lots and lots of prions that it can mess up.
Yeah. Okay.
Is that the definition of prion? Is it any kind of protein that has a tendency with some probability of misfolding at some point? Is that what a definition of a prion? Or is it a certain specific class of sequences?
I'm not sure. I'm not certain.
And does anybody know the mechanism by which a prion misfolded prion will cause other normally folded proteins to misfold and by contact? Does anybody say anything about the mechanism by which that happens?
That's got to be a high target of research, I would think, Eric.
Yeah, I would think so, yeah.
I mean, our awareness of prion diseases as opposed to more normal infectious diseases is really quite recent. I mean, you know, before mad cow disease showed up, and that was back in the 70s, the knowledge we had of guru or of the occasional sporadic genetically messed up what's called Creutzfeldt-Jakob disease after a couple of medical researchers. This was just not understood or well known at all. The mechanism wasn't really understood until they started investigating in the 70s.
I'd say just hacking out people's thyroid glands, lymph glands is not an adequate solution. I'm really still pissed off because when I was 12 and tonsillitis, they just hacked out my tonsils and adenoids because they didn't know what they did. So these stupid MDs didn't understand evolution at all, that things must have a purpose. They just hacked them out because they said, well, we don't know what they do, so they must not be important.
They were practicing medicine.
Yeah, there's got to be a better solution than just hacking out people's lymph nodes. They are valuable.
Well, I mean,
this is probably a good time to stop the discussion, but that sounds like a very interesting topic for another meeting, Eric. Thank you. And Jana, thank you for joining us. Would you like to introduce yourself before we end this meeting?
Hi there. Hi, I joined late, but it was so interesting and I'm eager to join the next one. So thank you all. I really appreciate it.
Yeah, that last one was fascinating. And I didn't get that on Stan's gone. He's playing pickleball or something. I didn't get a link to that one, but I would like to have that.
Carole, could you put that last one back into the chat again?
Oh God.
Is that the one that's 13 minutes?
No.
Okay.
Well, I'm not sure.
Okay, the last one is number 5.
Stan only posted 4.
Well,
okay, it's right here.
He posted a list of 4, and then he posted another one.
Okay.
Yes,
we'll add all 5 to the meetup description on Meetup in the comments. So if anybody wants to find out what the videos were, they can go look back in the Meetup, and it will be there before next week.
Yeah.
I'm not promising today, but we'll do it today if we can.
You can simply, get into your favorite browser and put videos, prion disease, and it will bring up a couple hundred of them. Or, videos, protein folding.
But it would be nice just to do that particular video, so
Yeah,
Yeah.
we'll try and put all 5 on the meetup description comments
Yes.
associated with today's meeting.
I don't know. I don't know what a meetup description comment, but
Okay.
I'll.
In the next 90 seconds, if you scroll up the chat, there is a chat from Stan that has the link.
yep, that's the link for number 5, the really long one.
Yeah. No, I found that. Yeah.
Number 5, 13 minutes. Then
Somebody was
above
using
that
terminology that was over my head.
are the 1, 2, 3, and 4.
Yep.
Just above that.
9:49 a.m.
We did not watch 4 because we ran out of time, and I really wanted to get to the prion diseases.
I'm going to watch it. I have nothing but time.
Okay.
Thank you all for coming.
Thank you, Carole.
Carole, are you okay with us posting the video for this meeting?
What?
Are you?
What?
We have tried to video the meeting that was not part of the presentations, not part of the videos. And is it okay for us to post that?
Sure.
Okay. So eventually this meeting will be on our list of videos that people can review.
Right, and that's why I put the video addresses on slides in the.
Thank you.
In the,
Now, is this going to be on do you have a channel, a YouTube channel that these?
Yes, the Minnesota Futurists have a YouTube channel, and we have about 10 of our recent meetings on the channel. And this will make its way to the channel over the next week, we hope.
Okay, that's awesome.
But now we've been having some trouble with, and we don't understand why, but some people don't have permission to watch the videos, and I didn't know that YouTube ever did that to people. Their point is to sell the videos by the advertising, and if you don't let people watch the videos, you don't do any advertising. So,
Or they're also into
David,
censorship for their religion.
since we do not record the videos,
and David,
I always put the addresses of the videos in my slides.
Thank you.
They don't want you taking Google ad money from the original presenters who did all the.
Yeah.
David, for whatever it's worth, I don't know if you have any of the original content from Brian Torin. He would put things on Zoom. If you have it, you could put those on YouTube too. If you don't have it, that's fine. But
at some point we may look into doing that. We probably want to talk to his family before we post anything like that.
Okay.
But
all right,
I'll let you know if we actually do some of that, because I think I do have a couple of his videos. At one point, I put together a package for his family of the ones we had.
But that's the reason that I always put the addresses to the videos in a slide of my
presentation,
my PowerPoint, so that people can actually watch the videos even though we don't record them.
Yeah. Thank you all for coming. Have a good week.
Bye-bye.
Thank you. Bye.
Thank you.
Carole,
Thank you.
great presentation. Thank you.
Great presentation.
Yeah, thank you, Carole.
That's wonderful. Thank you.
Good job. Thanks.
Bye-bye.
Oh.
Bye.
Yeah. David?
Yeah.
I see that I've gotten 2 messages from you that I haven't responded to. I'm sorry about that. Got a couple minutes now?
Yeah, actually I do.
What was up or not up.